Hi, it's Miss Kamitsi and Miss Along, and we're going to be helping you work through your notes on pages 10, 11, and 12 today. Where you left off taking notes before was we talked about nucleic acids and proteins. And they have an interesting relationship because nucleic acids actually have the code to build proteins. Your body builds hundreds of thousands of different proteins every day. Proteins like the structure of your hair and nails, the hormone insulin, antibodies, pigments like melanin, transport like hemoglobin. Your body is building these all the time. And what's going to be really important, remember, is the specific shape of the protein. If you look, this looks like a blob, but it's really much more than a blob. And where it becomes really important is where we talk about a specific group of proteins called enzymes. And so we're moving on to page 10 to talk about enzymes.
So with enzymes, there has to be some regulated conditions that are accomplished through the use of enzymes. We're going to be able to regulate some conditions. The enzymes will actually convert substrates, which are items that are going into the reaction, into products. That's the end result.
And so one thing is, the S in substrate could make us think of the word start, S-T-A-R-T. Enzymes are going to start by working on the substrate, and the product is what is produced.
Nia Golembi, come to the front office, please. Nia Golembi, if you're here, please come to the front office.
So enzymes are going to have four unique properties. The first is that enzymes are going to be able to speed up chemical reactions, such as synthesizing or making things, or digesting things, breaking them down.
So digest means to break down, and synthesize means to make. And enzymes can only do one or the other, but they'll accomplish both in the body.
They do this, they speed up these reactions by bringing the substrates, that starting point, together with the enzyme, and they're actually going to put them into an optimum orientation. This will reduce what's known as the activation energy, the amount of energy we need to start a reaction. And these enzymes are usually going to be proteins that are called organic catalysts.
So when we look at this section here, remember that the word optimum means best. Best, B-E-S-T. And so it's kind of like setting somebody up on a blind date, bringing two things together at just the right time. And that's what enzymes will do, is they'll arrange things in just the right order to allow a reaction to happen.
The second property is that enzymes are going to have a very specifically shaped active site. So in the picture, you can actually see the active site right here. And that's only going to fit with certain substrates.
And so some things as we go through, these four unique properties, the first is that they speed up reactions. The second is that they have a specifically shaped active site. That's the second property that Ms. DeLong said.
Our third property is that enzymes are going to be unchanged during the reaction, which really means that they can be reused over and over again.
And so you can see that property when you look at this picture. It looks kind of like a cycle. What we have at the beginning of the enzyme is how the enzyme is at the very end, which means that the enzyme can do its job over and over again. Notice, however, that the substrate is not the same at the end. And so we want to go ahead and fill in this chart. C represents the enzyme. And we would have known that this was the enzyme because it's unchanged. A represents the substrate. And we know this because this is what we start with, the S in substrate and the S in start. The products are what are produced. And this only happens because the substrate fits into the active site. And so the enzyme sped up this reaction because of this specific shape, and the enzyme is reusable. But there's a fourth property.
And the last property for enzymes is that they're going to work at their best, their optimum rate, in only some specific conditions. So if you change a condition like the pH, whether it's too acidic or basic, if you change the temperature, making it hotter or colder, or the salinity, changing the salts in the environment, then this can actually change the shape of the enzyme, which is called denaturing it. When you denature or change the shape of the enzyme, then the substrate, that starting point, won't fit anymore, and the reaction won't be able to occur.
So these are the three main environmental conditions. And so maybe let's add this word environment in here. If we change the environment, the enzyme won't work as well. It won't perform at its best. We're going to talk in just a second about pH, but you can see that changing the temperature is actually going to change the shape of the enzyme. And so that is really the fourth property, that enzymes can have their shape changed, and we really are going to have to practice this word. Now, if you look at the word denature, it could mean unnatural. So here's the native or natural state where we see the active site, and this enzyme will be able to do its job. If we change the environment, if we denature it, there's no active site, and the enzyme can't do its job because the substrate can't fit into it correctly. So, as we get ready to move on, just make sure that you notice the four properties. The first is that it speeds up reactions. The second,
That it's, that it has a specific shape.
the third, that it's unchanged and reusable.
And the fourth, that it has specific conditions or environments that will work at its best.
So we want to talk a little bit more about this word pH. And so we're going to talk about how the reaction rate of enzymes is highest in the optimum conditions of each organism. If you're a bacteria that lives in the thermal vents of the ocean, then you're used to heat and you perform best in heat. And if we put you in cold, you wouldn't perform very well there. However, if you are a penguin who is adapted to living in the Arctic, you're not going to work very well if we put you in the heat. So it all depends on that specific organism. And changing their optimum conditions can cause enzymes to denature, which can lead to death. We do have mechanisms in our cells that help us minimize these changes. And minimizing changes is also known as a word called homeostasis. You can almost think home stays. Homeostasis means that the internal conditions experience minimum changes. Kind of like your thermostat at home, which keeps your home at a certain temperature. Your body has ways to keep things from changing too much. Because if you look, if this is the optimum temperature right here, if this is the optimum temperature and we change the temperature, the enzyme does not work as well. So once you change the temperature, the amount of activity
Rosie Velasquez, meet your ride in the front office immediately. Rosie Velasquez, meet your ride in the front office now.
So to help maintain or minimize those changes, we have things called buffers that are going to help to balance or regulate the pH within a cell. Now, pH is actually a way to determine if something is acidic or basic. And there's a scale that's going to be able to help us identify which one it would be. On the pH scale, from 0 to 7 is going to be your acid. 7 itself is going to be neutral. And then higher than 7, so 14 is going to be your base. Now buffers will actually respond to the changes in pH and help maintain homeostasis and minimize any of those changes. And that's going to help those enzymes in your body that are performing all these different functions from denaturing, from having their shape changed.
So before we get ready to stop and write your summary, we want to think about all of this is about enzymes and how well enzymes work, and how if you change the environment, the enzymes won't work very well. Interestingly enough, acids can even be things like soda. So when you drink a soda or drink orange juice that has acid in it, you're changing the environment of your mouth, and the enzymes there could be in danger. But thankfully, your body has these buffers that regulate the pH. And pH is a scale, kind of like temperature. Temperature, we measure temperature on a scale. We measure pH on a scale. And pH really affects how well your enzymes work, which is why your body has a way to control that. Up next is your summary box. So make sure that when you answer your summary box, that you refer to a diagram and use at least 2 vocab words. And underline those words so that we can see them really clearly. Because we'll be doing an enzyme lab coming up soon, so we want to make sure that you've practiced enzymes.